So welcome to day 2. Hope everyone had an enjoyable evening. And I certainly think that yesterday was very productive and had some wonderful discussion, which we hope to continue today. As is sometimes the case, I've been asked to attempt a summary. I keep trying to see if other people would like to do this instead. That's what they say, and I don't believe that. But anyway, so here's a very brief summary of some of the themes that came up yesterday, and I apologize to anyone whose wonderful ideas are not captured here or are miscaptured. But Rebecca, if you could hit the next, So I begin with this idea, not because I don't think that all of you don't appreciate this concept, but it is important, and I think we're still sometimes drawn to the whole idea that, yes, of course, it's a community, and yes, there are interactions, but there must be an organism or a couple organisms that are doing yeoman's work here and are really, perhaps even in isolation, responsible for a lot of what we see. That could be true. And I don't mean to diss that approach, but it is much harder, of course, to think conceptually about units that are more complex, and then talk about how one attempts to either confirm or validate or refute the idea that this complex unit is in fact doing X. But I think that is where the big challenge and important area lies. We also talk about emergent properties. That's sometimes code word for we don't understand this yet, but perhaps there are conceptual theoretical reasons for thinking that some systems can't be necessarily predicted from their parts, but we'll see as to whether this is really something that is a major dominant notion or whether it's, again, just a question of time. Next bullet, please. We began with, and in fact continue talking about some competing forces or factors involved in a lot of the ecology and biology we talked about, where you could say that there are long-term evolutionary scale selective forces at play, and there's clear evidence of that, and probably 2/3 of all the presentations yesterday alluded to that or provided very compelling evidence of long-term adaptive, apparently adaptive changes that you would have predicted or perhaps explained by long-term selective forces. Then there are the things that we can observe in our own personal time scale and do and study in that way. And when we see what might be also adaptive changes that take place on that time scale, we think of them as undergoing or subject to selection as well. And you could imagine where, for example, in the case of antibiotics, there's been a decoupling of possible benefits that have accrued to a community over long periods of time in association with a host, and developing certain kinds of membership and properties and ways of transmission that are now separated from or at odds or worked against by these short-term pressures that are placed by an antibiotic with a kill effect when used in tonnage doses. So that's something that we talked about, and that is another one of those important issues that perhaps could talk about today as well. The next bullet has to do with assembly. There were a number of talks yesterday about how do these communities come together, and what are the factors that are responsible. Can we make some kind of headway in understanding and predicting and isolating these factors, different kinds of stories. And I believe that Brendan Bohannon, who is our first speaker this morning, may talk a little bit about this, and I would love to defer to him for a wiser discussion of some of these issues. But we in fact touched on a number of these points yesterday, whether there are deterministic or non-deterministic factors, and some of them could be historical contingencies, and some could be truly stochastic. The next bullet is related, but it has to do with timing. The importance of early founders and the effects they may have. Karen Gilliman talked a little bit about this in her talk, about the possibility of priority effects. Whoever gets there first has a different kind of role, can play a different kind of role than those that come after. There's an idea that some alluded to that my group has thought about with respect to whether some of these habitats could be viewed as open or closed with respect to their receptivity to outside organisms that are robbed by dispersal, and whether the habitat's even fully formed and all of the factors that might determine assembly truly determined at the time of birth or not. And I think in some cases, some habitats may be more so and others less so. But clearly a lot happens after birth, and there's a lot that we don't fully understand about the timing with respect to those events. And then Rick Blumberg in particular spoke about windows of susceptibility and opportunity and vulnerability and so forth with respect to postnatal timelines and even prenatal timelines in some of the other talks. The next bullet, I guess it's up there. Communities as part of a larger ecosystem, and I mean microbial communities. But we spoke a bit about, and Skip Virgin talked about, the interplay between genes and environment, where environment could be a whole variety of factors. And I threw in other because sometimes we think we're including, but really aren't, some of the other life forms, which clearly deserve a lot more attention. But they, too, alone are probably only part of the big story because we have all of these environmental exposures, which are hard to measure and even think about measuring, but a lot of them could have really important bearing on the history of the system and what it now is capable or not capable of doing or how it will respond now. So, for example, exposure to heavy metals or other kinds of xenobiotics that may have really important effects aren't being measured. And then finally, the last bullet are these big ecologic themes of stability and resilience and the whole idea of fitness landscapes and thinking about them as we try to design experiments and explain biology. Larry Forney has written and spoke a little bit about this. There are a lot of us that are trying to think in these ways because I think it's productive. There's a lot of great literature. Sometimes we end up rediscovering what's already been written about many times before over, in this case, 100 years or almost. And again, I think Brendan may touch on some of these themes. But the whole importance of interactions is acknowledged, and we talked a little bit about yesterday, but so important in even defining what is a community and what is rather just an assemblage or a random assortment of organisms. And we're, I think, beginning to learn about how to understand and measure and detect interactions, but there's so much more that is not being captured, especially with respect to community host or community environment interactions. And I've got two additional slides that bear on this, in part because Elaine Holmes was one of our scheduled speakers for this morning, and she would have been talking to us about the power of a technology for getting at some kinds of molecules that are important for interactions, and that's the metabolites, the small molecules that can be now measured so much more easily with both NMR and various mass spectroscopy methods. And she's got this tiered into different kinds of activities, starting with sort of measurement platforms and moving to analytical issues, kinds of data, and then finally algorithms and ways of processing and understanding information. And you could actually think about this more generically about other kinds of measurements and molecules or features that might also bear on interactions and how we would go about trying to integrate all of them. We also have a tendency to do two-way comparisons of different kinds of data. So, phylogenetic patterns versus metagenome patterns versus metabolomic or metaproteomic comparisons. And in the end, what we want to be doing is extracting features derived from all of these at the same time. And there are methods of doing that, and but they're not in wide use, and they still need to be further developed. And the last slide is another one of Elaine's slides that just, touches on some of the kinds of biology that her group and many others have thought. In fact, Marty began with some of this, that may be impacted by microbial communities within the human body. All my glasses out, you may or may not be able to read some of these different little features, but it's not truly important, except to suggest that there are many potential traditional human organ systems that we think listen to and respond to small molecules that are in part the result of microbial activities in the human body. Some of these are the products of co-metabolism, where there's a handoff of one molecule to the other, community to host, host to community, a further conversion or activity, and then a subsequent product is then acted upon. So it's complex, clearly, lots of possible places from which to measure these sorts of things, from fecal extracts to urine to plasma to any of the other body fluids and tissues. In fact, Jeremy sent a slide that I didn't put up because I wasn't quite sure it was real, but he's got a device that he says he's using in the operating room. Jeremy Nicholson's a surgeon, where the Bovie cautery gun actually has an import, a port that sucks in the fumes from the cauterization of human tissue, and those fumes go into one of his mass spec machines, and he's analyzing small molecules that are the product of, the aerosol, or the atomization from the material that he's cutting through. And he says that again, with Jeremy, sometimes you're not quite sure whether it's truth or fantasy, but he says that he's reading liver metabolites from Bovieing through a little bit of liver tissue and likewise other tissue. So you can only imagine that technology might at least spur the imagination as to how we get at some of these molecules and understand at least what's there as a starting point for understanding what's actually happening and what matters. So I think I will stop there and allow plenty of time for the other speakers this morning. They're all wonderful speakers and have wonderful topics to talk about. So I think with that, I will introduce our first speaker, or actually introduce Keita Bond, who will be the moderator for this first session. Sorry, Keita.
Not a problem,
Thank
David.
you.
And I just want to add sort of my take to some of your points. Here are sort of the bullet points I heard yesterday. Microbiota contains multitudes, a la Skip. It's not just the bacteria, it's the viruses, it's the fungi, the protozoa, and the worms. And beautifully co-evolved to be important allies and sometimes foes. This microbiota plays important functions in energy regulation, in immune system development, in organ function. That location and structure matter, and similar populations have different functions in different sites, like the oral cavity versus the vagina, and that structure, such as the mucin story, really can make a big difference. That model systems from trees to insects to animals to man all are contributing to this very complex slide that you just showed up. What are the signals and how are these communications all taking place? And we're learning different parts of the story from these different models. That changes in our ecology, such as the use of antibiotics and clean water, are really making a difference in changing disease patterns. And the whole obesity, the asthma, and the changing patterns of cancer were really dramatic to me. It's not just the microbiota, as you just said, it's the host, that the genetics of the host really determine who gets in to some extent, and that the environment also is playing this very major role in also contributing. And the issue that you raised about timing and the criticality of that window of opportunity that seems to be there. And so one of the things as a policy person that I'd love to have some discussion about as we go along is one of my observations is the changing structure of the labs that are being displayed by the various speakers. They're very large. They have a lot of different skills, and I wonder if that means that we should be supporting this kind of science in new ways, and I worry about academic recognition for the scientists who are doing this work. And the question you raised, David, about what kinds of technology are going to be needed in the future, I think is also a very interesting point. And finally, the one thing that I took away from Marty Blazer's talk is, are we all going to be getting, along with our vaccines, at 2 months a dose of synthetic poop in order to make sure all our immune systems develop properly, that the gut functions are well established, and the energy regulation for the rest of our life is going to be appropriately set up, a la Lauren's question.